and he's going to tell us about temporal dynamics and nucleation of ERA, phase-separated condensates, and estrogen signaling.
So good afternoon to all. I am Rajat from Dr. Dimple Notani's lab at NCBS. And first of all, I would like to thank Anjana for this opportunity. I'm going to talk about the temporal dynamics of this protein called estrogen receptor alpha, and which is in regulation of estrogen signaling. So before I start, let me give you a brief overview of the signaling system that we work with. Estrogen is a steroid hormone, and being the steroid hormone, it can translocate through the plasma membrane inside the cytoplasm, where it encounters its receptor called the estrogen receptor alpha. And upon binding to the receptor dimerizes. And upon dimerization, what happens is it translocates inside the nucleus, where it binds to different E2 regulated genes, the estrogen regulated genes. And that is how it regulates the transcriptional landscape of the cell. So while working on the system, us and others in the field have found that estrogen ER alpha as a protein forms these condensates, or these foci once E2 is given to the cell. And what we see is initially there is a growth in the number of these condensates, which is plotted as the number density. The number of these condensates increase with time after you give the ligand, which is E2. And after a certain time, we have found that this sort of reaches like a stationary phase, and it doesn't increase much after that. But why exactly are we interested in these condensates is that we have seen in a study of ours is that the peaks that you see are the estrogen binding, are the ER alpha binding profiles in the genome. So these are two different estrogen responsive genes called TFF1 and CCND1. What we have found is that these responsive genes have propensity of binding of ER alpha in clustered manner. And where exactly in these clustered regions, we see a lot of polymerase and other activation machineries. And these activation machineries are important for the transcription of the cell. And because of this sort of a clustered binding, and if we somehow, using CRISPR or any other techniques, if we can somehow perturb this clustered binding, what we have seen is the formation of these spots using immunofish techniques, that the formation of these condensates also gets affected. So what we think is that these condensates are these dynamic loci of multiple of these clusters coming together and creating like a active environment for the transcription of the E2 responsive genes. So this is the overall E2 program that is regulated by these condensates. But this E2 regulated program is very time-driven. There are certain genes that need to be transcribed initially, and then there are late responsive genes as well. So the formation of these condensates has to be in a time-dependent manner. There should be certain regions that need to grow faster, and there should be certain regions that would take its time. So we again took the same thing. So the overall question that I'm trying to address in this talk would be, what decides where the condensates are formed first? What are these all specific regions where these condensates are being formed first? So before getting into that, let me tell you how ER alpha looks like. So ER alpha is this traffic pole-like structure is not exactly, this is the very crude diagram of how ER alpha is. It has a ligand binding domain, the RNA binding region, which is in the hinge region, and a DNA binding domain. So it has both the ability to bind to RNA as well as DNA. And this RNA binding property of ER alpha has already been shown by 2 studies from our lab, that it tends to affect the recruitment of condensate at different locations. So what we would find is that these condensates would be sort of mixed of two, like there can be some RNA-bound condensates as well as chromatin-bound and freely floating as well. To tackle that problem, what we created is we created different mutants, and we created RNA-binding mutants, the mutants which no longer have the ability to bind to RNA, so it would just bind the DNA. And we created P-box mutant, which mutates the DNA binding region of ER alpha, so it would just bind the RNA, and a double mutant in which both the DNA and the RNA binding domains are mutated. Things that are pretty obvious from here, that the RNA binding, the mutants that is only able to bind to RNA tends to move faster as compared to the ones that are only able to bind to DNA. To look at that, what we did is we checked the trajectories of these condensates in the wild type and the RNA binding mutant. We can just bind to DNA, and the DNA binding mutant. And what we did is we took these different trajectories and plotted a survival plot of this. This is the fraction of the particles that are there at these with different track lengths. So what we see is the in the RRGG mutant, which is just the DNA binding mutant, we see that the fraction of the particles that have a higher track length is more, which could be seen from the trajectories as well. So now we have a system in which we can differentiate the different sort of condensates that are there. In the P-box mutant, we don't see the formation, but in the DNA binding mutant, we see that these sort of condensates are very mobile and they move a lot, and so we have a shorter trajectory. And in the RNA binding mutant, which can just bind to the DNA, so those tend to stay they have a higher track length. So now I would try to reiterate the question is, what decides the DNA-associated condensates that are formed first? So because we want to see where exactly in the genome and what exactly determines these factors. So we went back to genomics, and ER alpha is the transcription factors. So the first thing that comes to mind is wherever there is a better motif. So this transcription factor would bind to a given motif. So wherever there would be a better motif, those should be the regions that should ideally bind ER alpha more. But once we looked at the motif, so what you see on the y-axis is the motif score. The best the motif, the higher the score. And this is the signal that is there, the signal of ER alpha. So the clustered sites are the one that I already talked about, the ones that are actually contributing to the formation of these condensates. And the non-cluster sites are the ones that don't contribute there. So what we find is that with the ER alpha motif, with just the basic information of what is within the DNA, we are not able to differentiate if the particular, if the signal at that particular ER alpha bound sites. So now we looked at, we try to differentiate what exactly differentiate the clusters that contribute to the formation of ER alpha condensates versus that don't. We found that there is a certain thing called as FOXA1. FOXA1 is a known pioneering factor and is known to interact with ER alpha as well. And what we find is that for the highest binding sites, which you see here, for the highest binding sites, the FOXA1 motifs tends to be better as compared to the non-cluster sites, which don't contribute. And let me give you a brief overview of what FOXA1 does. Like I said, it's a pioneering factor. What pioneering factor does it can bind to close chromatin. And upon binding to the close chromatin, it has a domain which can innervate inside the nucleosome and tend to remove the linker histone. And then it can also recruit other chromatin remodelers, which can open up the DNA. And because of opening up of this DNA, now the transcription factor, which is ER alpha, and other transcription factors can also bind in. And so it can be possible that FOXA1 is trying to create these sites even before signaling, and that is where ER alpha can, so FOXA1 sets the framework, and then ER alpha can come in, and then the signaling would proceed. So what we did is we looked at the clusters that have FOXA1 even before the signaling started, and then, so what you see here is time course ChIP-seq of ER alpha. So like I was talking about the binding sites of ER alpha, now we are plotting that how much signal is on those binding sites with time. And what we see here is the clusters that have FOXA1 tend to gain the binding of ER alpha faster. So genomically, wherever there is FOXA1, even in the -E2 condition, wherever there is this pioneering factor in -E2 condition, those regions tend to gain ER alpha faster as compared to the ones that don't. Now this is all genomic. Now we wanted to see if this is true for the condensates that we see as well. So first of all, what we try to understand if there is some sort of a deposition to regions that have that form condensate, is it not a uniform distribution? So what you see here is in the first 10 minutes, where exactly the condensates are forming. These are overlaid in the first 10 minutes in 3 different cells. And what we see here is that there are certain regions that are more propense to form condensates, and which is compared to the uniform distribution which you see here with the same number density. So this was done along with Madan. And what we wanted to see if the regions that are sort of showing a higher propensity to form these condensates, if they have FOXA1. So right now this is a work in progress. What we have done is we have looked at FOXA1 in the same condition with ER alpha. So what you are seeing is the mutant that is only able to bind to DNA. And what we see here is that in the initial part of the signaling, we see that the first sort of condensates that are forming, so we are still quantifying the same, that in the initial part, if the condensates that are formed first, that form on these FOXA1 rich regions. So this is true for the FOXA1 rich regions, but now we wanted to see if you look at any E2 responsive genes, so because in the end, the goal of ER alpha is to stimulate the E2 responsive gene. So we looked at one such gene and performed FISH with that. So as already talked by the previous speaker, thank you for that. We performed immunos-FISH of NRIP1, which is an E2 responsive gene with ER alpha and FOXA1. And we looked at the intensity at 0 minutes when you have not given the signaling, and at 60 minutes when you have given the signaling. And what we see is the normalized intensity at the FISH spot. And we see that the FOXA1 tend to be more present at the NRIP1 spot initially at 0 minutes as compared to ER alpha, but as the signaling proceeds, the amount of ER alpha at those regions increase. So in the E2 responsive genes, we see that the overall FOXA1 is present at first, and then ER alpha comes in. So we thought what would happen if you remove FOXA1 from the picture. So we treated the cells, we knocked FOXA1 down, and what we see is we looked at the ER alpha condensate formation, and what we see is now, which is quantified here using the volume fraction, we see the formation of condensates gets affected. So once you remove this particular pioneering factor from the picture, we don't see the formation of these condensates. So this is still a work in progress right now. We are trying to see if the regions that are there that show a higher FOXA1 rich region, those show up bigger trajectories or not. We are trying to figure it out. So this is the take-home message what we have right now, concrete, that the DNA-bound ER alpha condensates tend to display longer trajectories as compared to the RNA-associated one, which are present in the P-box mutant. The FOXA1 motif shows a higher prevalence in the clustered sites versus the non-clustered sites, which do not contribute to the formation of these condensates. And the FOXA1 pre-bound condensate clusters tend to show a faster growth, which was shown here. And then if you look at the, using the FISH, what we have seen is the FOXA1 tend to remain bound at the E2 responsive gene even before signaling, and then it grows with time. And finally, if you remove FOXA1, then we don't see ER alpha condensates at all. So this is the model that initially there is some sort of a pioneering factor, which is FOXA1, and which nucleates or opens up the DNA initially, and then as soon as estrogen comes in, which translocates and this form these condensates. With that, I would like to thank my supervisor, Dimple, and the collaborator, Jyotsna and Madan, and the lab members, Dipanshu, Zubair, and others, and funding agencies. Thank you.
Great. Thank you very much. Questions for Rajat?
Nice talk, Rajat. I have a question. So this FOXA1 site and the ERE, how close or how far they are, like in base pairs?
Within the motif, you have like a distribution. I don't exactly know the distance on top of my mind.
But is there a double peak? That means those who are regulated or affected by the FOXA1 are close by, and then the other ones are far away. So I would
within the peak, I am not really sure of. Oh. Within the peak, how the distance looks like, I'm not really sure of that.
You got a microphone.
Rajat, that was interesting. It still doesn't solve the chicken and egg problem because something has to get FOXA1 there.
So initially what we think for these particular few of these motifs, there is a FOXA1 motif near the very highly responsive clusters. So the FOXA1 motif can come there, and that is what sets up this thing. And from once the FOXA1 comes and binds and opens up, then the other transcription factors can come. So the motif is already there for FOXA1.
And the idea is that the FOXA1 is usually a long-lived binder, that the turnover is...
Yeah, so what we think is that the FOXA1 can... So for the... I'm not sure about the dwell time of FOXA1s on the chromatin there, how different it is for pioneering factors versus others, but that's the overall plan that tends to come and bind in the close. So people have talked about that the binding in the closed chromatin state, it is there, but how long and how different it is from open.
Nice talk. Is there anything else other than FOXA1, you think?
There are a lot of pioneering factors that are associated with ER alpha. So yeah, there is GATA.
They would all be there in these condensates, or
what exactly?
You think they would all be there in the given this?
So there are papers that have talked about this, but not all. I'm not really sure of if all the condensates would have all of these. So with ChIP-seq experiments, which is like a snapshot of multiple cells, like millions of cells together, people talk about so many of these factors being together, but I'm not really sure if every single one of them would have every single one.
How many factors are you talking into?
Like, so roughly? I'm not really sure. I can't put a number on
range of tens or?
So So there is a subset of subset of these genomic regions called the megatrans complex that have like a complex of like 10, 12 transcription factors coming together. But will these condensates would have all of these? I'm not really sure. Not sure.
What's unique about the genes that have the FOX sites? That why do they need to be driven? Is it, sequential importance or temporal importance, or what can you say about
So in terms of so if you look at the FOXO1 motif and if you look at the overall binding strengths, so how much binding is there and if there is a presence or absence of FOXO1 motif, we see that the very highly binding sites tend to have FOXO1.
Right, but I mean, what's special about those genes? Why do they need to be turned on?
So in terms of the early or late
Yeah.
responsiveness of those genes, I don't know.
Thank you very much.
Thank you.